And then we also saw that the cells really started to align, like we previously said. They aligned around in the circumferential direction around the ventricle. So that got us really excited. And the next step there was then to take this alignment data. We know that they've aligned. And we also can visibly see that they're contracting. So are they contracting in the direction of that alignment? Which is what we hope to see. And so in order to kind of investigate that, we use calcium imaging, which is really just staining for the calcium activity. We have this marker that stains for the calcium activity inside cells. And so when cells contract, they really cycle calcium very rapidly. And this cycling of calcium we use it as a visual indicator for contractile activity. And what's really cool is that if you kind of watch some of the videos that are posted online, you can see this calcium wave propagation that indicates that cells are contracting in the direction of their alignment. They kind of have this nice even coverage around the ventricles, and they spontaneously contract over time. And so that was encouraging for us, and we then moved on to looking at, okay, can we now control this contraction? Can we stimulate the entire ventricle, and can we get it to contract all at once? And we were able to do that very successfully at different frequencies. We were also able to measure out how fast are these calcium waves moving. So how fast are the conduction velocities of these cells. So those were very interesting experiments that were nicely outlined in the paper. And then the next step to kind of investigating the function of these ventricles is to see, can we start to replicate some of the native functions of the heart, right? So we know that the ventricle in the body really its function is to pump out blood by restricting volume of the ventricle. So contracting so that you reduce volume of the ventricle, and therefore you're ejecting fluid from inside the ventricle. So we want to see if that volume decreased with respect to our printed ventricle. So we did a very simple study by just looking at the opening, the diameter of the opening of that ventricle. So did it decrease in area over time, over different contractile cycles? And we found that it did. And also we saw that what was really interesting was that we saw also wall thickening. So during contraction, your cardiomyocytes contract, and then in the other direction, they thicken. And so that is a hallmark of contraction in the native heart. So we also saw that as well. So I think taking all of what I just said together, these were good indicators that we are moving in the right direction, that we're kind of making this model that is able to replicate some of the things that you find inside your heart, for example. And there's obviously a lot of studies that need to be done on top of that, but we're doing these first steps that really are getting us to a starting point for a model.